I'll probably just do a little video on purine regulation before I post it. Uh, so again, if you don't want your face on it, you can turn off your camera for today. But let me just continue. So our purine salvage pathway. So our purine salvage pathway is one way that we can actually salvage some purines, right? So this can actually go, we can actually form only IMP and GMP, not AMP. So that's a big thing. It's only IMP or GMP. So we go through this HGPRT, which is a horrible name for an enzyme. It's our hypoxanthine guanine phosphoryl transferase. It's a mouthful. So just HGPRT, right? So these salvage purines from RNA and DNA degradation, right? So as I said, like the big thing here is like adenine is not salvaged by HGPRT. Okay. Um, so the most abundant purines, and well, adenine is one thing. It's like the only reason adenine is really not salvaged is because if we keep salvaging so much adenine, we're actually going to be blocking IMP and GMP formation because it's already one of the most abundant purines in our bodies. And if we keep salvaging it from our degradation of our RNAs and DNAs, we're going to start blocking different pathways, right? Similar to what we did here, or like, for example, here, or how we look at it like forming AMP and it blocks the whole pathway, right? If we keep salvaging so much adenine, we're actually going to be blocking our pathways indirectly as well. But the good thing is we do have a way to salvage some of our adenine, which is through our adenine deaminase, which converts adenine to hypoxanthine, which is also really important in, for example, reforming PRPP into IMP. Okay. Um, so this is kind of like one way that we can actually salvage a lot of the purines that we get through, digestion, through protein turnovers and stuff like that. So this is your more medically relevant stuff. So gout. So gout is actually an excess of uric acid. Okay. So you're already seeing a lot of these different chemicals, our GMPs, our EMPs, our xanthines, right? So this is gout is usually treated with allopurinol, which actually inhibits our xanthine oxidase inhibits our xanthine oxidase. So it's a xanthine oxidase inhibitor. Sorry, that sounded a little bit confusing right there in the slide. It's a xanthine oxidase inhibitor, right? Um, so these actually produce an excess of hypoxanthine and xanthine, which is just harmless and it's excreted in our urine. Okay. So we had the purine salvage pathway, and this is our purine degradation pathway. Any questions on purine? I know that was a lot of information right there. The good thing is, like, for the purine thing, for some reason I struggled a lot with it when I was taking this class, and if I remember I know for a fact he's gonna ask at least one gout question. Like at least he's gonna ask like, what does allopurinol inhibits, or what is the medication for gout? So gout is a big important thing, but and just knowing that the purine salvage pathway, we don't form adenine. That's like a big idea, right? Because a lot of people will say it salvages AMP and GMP, but no, it's only GMP and IMP, which is also okay, right? Because IMP is still a metabolic crossroads, so it can form AMP if we desperately need it to. Okay, so now we're just gonna jump into Xylee stuff. So I think you guys noticed already if you've gone through some of his lectures, Xylee doesn't like writing. I don't know what is his war with slides with writing, but that man just likes talking in images, okay? So that's why it's kind of annoying. The good thing is I did go through all his lectures, so I think I got most of his big ideas for each of his lectures written down here. So at least you got that. So the first thing we talked about in lecture 21 is lipids, right? So lipids are pretty important, right? We can use it for energy storage as adipocytes or adipose structures like cell membranes and signaling coenzymes and pigments, and the important ones are like hormones, right? So these are cholesterol-derived hormones. So these are really, really important, right? Cholesterol is important for a lot of different hormones like estrogen, cortisol, cortisone, and prostaglandin is also really, really important signaling. And I don't know why, but the only thing that's reminding me of like what prostaglandin does is like childbirth. It's really important in childbirth. I don't know why that just came to my head, but it's really important. Wax here, on the other hand, as you know, is really important for structural integrity as well for different plants, animals, earwax, for example. Uh, but the important thing about lipids is our fatty acids, because fatty acids are going to be important, especially when we talk about cell membranes. Okay, so we can have saturated and unsaturated. Okay, so for the saturated, these are the saturated means that they don't have any double bonds. Okay, so important properties of these saturated fatty acids that the larger the carbon chain, the higher the melting point will be. Okay. For unsaturated is when we actually have a double bond in our fatty acid. Okay. One or more. And the larger the carbon chain in an unsaturated, the lower the melting point is going to be. And the pi bond will always be in a sort of a cis conformation. So the fatty acid molecules are always going to be pointed in the same 3D orientation. Okay. That's just a little detail I don't think he even talked about. I just remember that from one of my classes, but I think it's like just important to you to visualize how these pi bonds or how these double bonds are affecting our saturated acids. Okay, so I do have a question really quickly. So which one of these will have the lowest melting points?
Palmitoyl.
Yeah, I got it.
The
Yeah.
palm acid.
Yep. I love how Jaime just put like, I'm just gonna put the like the nomenclature, like the signaling that there's a double bond there. Yes, so the one that has a double bond is gonna have the lowest one because the double bond is there. Okay. So I know we're gonna probably gonna talk. I think I remember putting in a slide a little bit later, but the important thing is like when you think of a double bond, think of a kink. Think of an error in the structure, in a whole structure. Okay, especially because fatty acids are gonna be like layered on top of each other. If there's a double bond, where you're creating a crack in that layer. Okay, so it's kind of an integral weakness, and melting point is just a way that we can like determine if something's structurally sound or not, right? If it's a high melting point, it means the bonds are super strong with each other. If it's a low melting point, it means it's not as strong, and that's where these pi bonds come in because they're like a little crack in the whole structure. So triacylglycerols, these are the really important for energy production and like heat production as well. So these are actually three fatty acids in an ester linkage to glycerol. Very important. It's an ester linkage. That's like a very key thing that you should know. It's an ester linkage. These are also nonpolar and hydrophobic. Now just jump in a little to more cell relevant stuff for or for fats or lipids. I mean, we have two very important structural lipids. We have our glycerophospholipids and our sphingnophospholipids. So you should know these properties, these differences between both of them, because I've seen him ask on exams. So glycerophospholipids has two fatty acids. Its head group is an alcohol and has an ester linkage. A sphingolipid is one fatty acid. It has one sphingosine group, and its head group is polar. It can either be glycosidic or phosphodiester, and it has an amide linkage. The big important properties to know here is the difference in fatty acids between both and their linkages. Again, glycerophospholipids have an ester linkage, while sphingolipids have a amide linkage. Okay. Now going to why this is important for a cell. We have something called our glycocalyx. These are just areas that are really important for a multitude of reasons, such as tissue and organ specificity, cell recognition and adhesion, cell migration, blood clotting, immune response, wound healing, and nerve impulse and transmission. And you're going to see that these areas are going to have very different levels of certain structural lipids. And you can't really think that a structural lipid is going to do the same thing, okay? Because they all have different properties and they're all going to be used in a different manner, okay? So now that I just told you guys what they are, which one of these is our glycerophospholipids and which one is our sphingolipids?
The one with the phosphate group is our phospholipid.
Yep. Can anybody tell me why the left one is our sphingolipid? See, our left one here is a sphingolipid because we have an amide linkage. You see that nitrogen right there, creating that amide linkage. So that's like one big way of like knowing it. It's based on what they're bonded to. In this case right here, we're actually having an oh here right here, and we actually have an ester linkage right here, as we see right here, right? A double bonded O and another alkoxy oxygen or an alkoxy bond. These are like ester linkages, right? And this is an amide linkage. That's how I usually think about it because you're never gonna see nitrogens. I'm not gonna say never, but for the most part, you're not gonna see a nitrogen in a glycerophospholipid. In a sphingosine, it's a little bit different because a sphingosine has this amide linkage, so it has to have a nitrogen in there somehow. Any questions on lecture 21?
Are sphingos, however you pronounce it, are they always gonna be unsaturated because the sphingosine part of it has the double bond?
Yeah, but when I think you're like more referring to like saturated and unsaturated in just fatty acids, right? In general, like their properties.
All right,
Um,
it's not gonna
yeah,
get like
it's not
that.
gonna it's not gonna matter that much because we're not looking, for example, um, it just matters a lot more with glycerol phospholipids just because that's what we're gonna see in cell membranes. And Dr. Zhao is just trying to prime you to what he's gonna show you in the next couple of lectures, right?
All right,
To be honest with you, this is the last time we're probably going to talk about sphingolipids, if I remember correctly, for the rest of this class.
all right,
It's
fair enough.
just yeah, it's just so you know a structure, one other structural lipid. Yeah. Any other questions on lipids? Yeah. So introduction to our biological membranes. So cell membranes, very important, right? Protects us, keeps our cells intact. So these are going to be made mostly of lipids and proteins, and there are going to still be some glycosidic or glycolipids, glycoproteins. So there is going to be some carbohydrates in there, but for the most part, lipids and proteins. So the biological membrane has three important properties, or up six or seven important properties. So we kind of just put it as College of Medicine death as a way to memorize it. So cell-to-cell communication, organization, reaction pathway, molecular traffic, divide cells and organelles, energy transduction, boundary and transporter receptor, and adhesion molecules. So again, the mnemonic that we kind of use is our ComDept or College of Medicine Det as a way of trying to memorize these. Can anybody tell me what are the three characteristics of a biological membrane?
Semi-permeable.
You got this, Matt.
The the mosaic, and would you just say lipid bilayer?
Yeah, I think those are because I also forgot completely, so that's why I was like hoping one of you guys remember what he said there. Hey, you can't say I'm not honest. I wrote these slides a couple of months ago, but I'm fairly sure those were like at least those first two were completely correct. But the important thing here is the plasma membrane symmetrical or asymmetrical?
Asymmetrical.
Exactly. And that's really important for functionality of cell. Because some cells, especially if we're looking at a cell that's connected to the lumen of some, are either our alimentary canal, our nephrons, the cells that are closest to these lumens are going to have a lot of different proteins than the other sides that are not touching that lumen, right? So they're considered asymmetrical depending on their functionality, depending on what they're doing on certain areas of space around the cell, right? These membrane proteins are the ones that are moving everything around, communication, transporting, right? So there's three different classes of membrane proteins. We have our integral membrane proteins. So these can be either monotopic or polytopic. Monotopic meaning they only go through one membrane, either the inner or the outer, and polytopic meaning they cross both. Our peripheral ones are the ones that are not connected to our membrane, but they're associated with it through electrostatic and hydrogen bonding. So they kind of float around the cell. Okay. And then we have our amphoteric proteins. Okay, so these can actually interact reversibly with our membranes. Okay, and they usually have some modifications for the proteins, which is either lipidation, phosphorylation, or ligand binding. Okay, so as you see right here, like right here in the top, we have our GPI anchored protein. Okay, and they can actually move around. They can either attach when they need to and deattach when they don't need to as well, right? While peripheral, like the difference I think a lot of some people have is the difference between peripheral and amphoteric, because that peripheral is like almost like an orbiting, like our moon, right? It's orbiting the cell. It might not always be interacting with something, but it's always around the cell moving, okay? The important thing here is our hydropathy index, right? So can anybody tell me what this is trying to tell us?
How much it really doesn't want to be near water.
Basically. Yeah, so it's basically telling us how certain amino acids are gonna interact with water. Is it gonna be in the inside of the cell, inside of that cell membrane where there is no water, or is it gonna be the inside of the cell or outside of the cell where there is gonna be water, right? So you're gonna see right here that a negative delta G, it means it's hydrophilic, it wants water, and a positive delta G is gonna be hydrophobic, okay? So for a hydropathy plot, this is also a really good way of representing what's happening with a certain protein associated with a membrane. So if we follow along this graph, you're going to see that a lot of these, as you're going to see that a lot of these proteins are in the hydrophilic side, so they're either in the in or the out of the membrane. But this right area right here, this big spot right here in the hydrophobic side is going to be in the inside of our cell membrane, okay? So this is just one way, and you're going to see that these numbers are just representing different amino acid residues of a bigger protein. Okay. Do I have a question now? The
I have a
big.
question actually.
Oh, wait a second. Let me just
Can I
finish
ask
the
you
thought.
real? Okay.
Yeah. So the big important thing for this is knowing what you're seeing right here. Hydrophobic means it's going to be the inside of that cell membrane. Okay. And I trust like there is going to be a question on these hydropathy plots, and they're just going to ask how many protein residues are going to be crossing into the cell membrane. Okay, so you're just going to have to count the big hills that are in the hydrophobic side of this graph. And I know that there are some little ones right here, and you're probably asking like, oh, how am I going to differentiate between both of them? They're going to make it really obvious which ones are on, like they're not going to make these kind of like, ooh, maybe it is, maybe it's not. No, it's going to be like big hills. Okay. Matt, what was your question?
The I don't know if units is the right word. The box that you've got, the hydropathy
Okay.
index, is that is delta G?
Yeah.
Those numbers?
Yeah, it's just representing delta G. Yeah. I don't know why they didn't put the units in the top with the hydropathy, but yeah, I'm fairly sure it's delta G. If I remember correctly, it's delta G.
Okay, thanks.
Yeah.
Just really quickly, if we're going to have to potentially be counting how many residues would be like hydrophobic, can you?
Oh, it's not gonna be residues. It's gonna be like how many times does the protein cross the cell membrane? Because when
Okay.
you cross to one side or the other, you're gonna be going through the middle, right? And those
Okay.
are gonna be our hydrophobic residues. Are gonna be the ones crossing into it, if that makes sense.
So for example, with the one that you have here, what would that answer be?
Just once.
This one. Okay, that's what
Yeah.
I'm seeing, but I just want to make sure.
Yeah, but like they're gonna be like, because I hate this example for some reasons. I didn't put a clear example, but for the last couple of years, when I've seen like when I've heard how the example, it was like pretty in your face which ones were the hydrophobic. It wasn't like, ooh, like this one kind of crossed, maybe it's not. No, it's like really in your face, because if they try to confuse you, that's gonna be kind of a pain, right? That like.
Yeah, because like in the lecture, he was talking about like there's 20 residues. And so I've been thinking that I'm counting the number of residues of that one big hill, you know?
No, yeah, but like you can still count. I just don't remember if he did counting or not. He did counting on my exam, but he hasn't done it in the last 2 years. But you can still kind of get an idea just looking at it and just seeing is like, because again, they're not gonna make it hard. They're not gonna make it like residues that cross 70, 71, or 72.
Yeah, I mean, like just by looking at it, you can tell it's like 75 to 95. So,
Exactly. But
okay, so it might be a question like that on the exam.
there might, I just haven't heard of it in the last the last 2 semesters. I haven't heard him do it. The more important thing to understand is just knowing which ones are the ones crossing. And then if you have to count, it's usually going to be like a ruler, you know. You're going to see it starts at 70 and it ends at 90, so you can kind of approximate a number, and they're not going to make the number super close. All right. So just jumping in. So there are some special rules for where these amino acids are going to be in regards to the cell membrane. The positives are usually going to be inside the cell. Okay, so our positive amino acids, lysine, histidine, arginine. And then tyrosines and tryptophans are actually going to be located right in that membrane interface before entering the bilayer, right? So they're going to be right at the door, but never cross the door. Okay. So you see kind of like right here, our tryptophan and tyrosines never are in the middle, or rarely not in the middle. They're mostly either like at the doorsteps of the bilayer. Any questions on this lecture? Lecture is 23. So now that's when our fatty acids and those unsaturated unsaturators are going to come in, David. This lecture, our membrane proteins. So how does temperature impact our membranes? So for the most part, our membranes are in this kind of equilibrium between disorder and ordered states. So our L0 or our LO and our LD, okay? So it's in this equilibrium. So if LO is our more rigid membrane and our LD is our more fluid membrane, if we start decreasing from our melting point, our temperature, we're going to be in a more ordered state, right? As we freeze, right, our membranes are going to be in a more ordered state. As we increase our temperature, we're going to start going towards this disordered state, okay? So how is this important for unsaturated and saturated, right? So we talked about how unsaturated fatty acids kind of make a kink, right? So once we make a kink, we're going to have a lower melting point because the structure isn't ordered as much as it could anymore, okay? So that's like the big thing. If we have an unsaturated fatty acid, we're going to have a kink, which means we're going to have a lower melting point, which means we're going to be a lot quicker to reach that disordered state than it would be with a saturated fatty acid. Does that make sense? Okay. There is going to be a very important question that's going to come up, and I swear to you, Xylee always asks this one question, so I always prepare my students for it. But before I talk about this, the other big important thing is that membrane proteins, our phospholipids, can move around very easily laterally, very fast. Transbilayer is kind of harder. Okay, so if we didn't have an enzyme, it would take usually half a day almost, or I mean, half of the half reaction of a flip-flop movement would be days, right? So we know that doesn't happen. So we know there is an enzyme that works with it. It's flippase, floppase, and scramblase. Okay. So flippase actually moves our phospholipid in to the membrane, and then our floppase goes out of the cell. Okay. So again, flip I goes in, O goes out. Okay. So they're both using ATP. Okay. And our scramblase just move in either direction depending on where the equilibrium is. So the big also important thing to know is that flippase is a P-type ATPase, okay? While our floppase is an ABC transporter, okay? This is gonna come up a little bit later, but this is pretty important as well to at least know. So, okay. So this is the question that always comes up in an exam, okay? So polar bears are unique animals that are able to withstand extremely cold temperatures due to their thick coats. When looking at their cell membrane composition of these animals, what could you expect to see in regards of the amounts of saturated and unsaturated, okay? So I'll just give you guys some time to think about it. The big thing here is that word extreme cold temperature. What happens to fatty acids in cold temperatures, right? They go to ordered state a lot faster. And we want that equilibrium, right, between order and disorder. So actually Matt got it right. So Matt, what was your thought rationale on this?
That nothing is all or nothing. And so it needs to have more unsaturated, but it's not going to just be only unsaturated.
Well, yeah, we need more unsaturated to form a kink, so it's not constantly in that order state, right?
Yeah, I mean, if it's going to be crazy cold, then something that would be more flexible at like very flexible at a normal temperature is going to be around the
Yeah.
necessary rigidity at cold temperatures.
Exactly. So that unsaturated is just adding some disorder, so it's not constantly in that order state where we don't want it mostly in that order state. We want right in the middle. So creating that disorder kind of breaks apart that order state, so we go back towards that middle. There's always a question on the exam, and it's always some animal in either a hot environment or a cold environment, and then he's gonna ask what's the ratio, or like what do you expect the amount of saturated to unsaturated fatty acids. So understand that process, right? So let's say that we're talking about what's an animal in a hot temperature. I don't know, let's say it's a gorilla in a really hot environment in the world. Okay. So in that case, we're gonna most likely, if there was no difference in saturated and unsaturated, there's gonna be a higher amount of cell membranes in the disordered state, right? Because it's hot. So if it's in the disorder state, we want more saturated to bring back to that rigidity that we need to maintain for a cell membrane, right? So there's always questions like that. So just be aware. This is just a little practice. I think this came from my exam, actually. So I think I just rewrote it to work for this PowerPoint. So trust me, there's always an exam on this, or there's always a question on this on the exam. Okay. Any questions on this? No. Lipid rats, or lipid rats, I mean rafts, sorry. I've been working so much today that I am exhausted. I can't wait to take a shower and go to bed. Okay, lipid rats, the rafts. Oh my God, lipid rafts. So these are outer leaflets. So these are the cell membranes that are outside of the cell. So these actually contain sphingolipids. Again, probably the last time you're ever going to hear about this. Sphingolipids, cholesterols, and GP-linked proteins. So they're actually areas of our cell membrane that are slightly thicker and they're more ordered than normal. Okay, and this is important because these are actually areas that scientists might not know exactly what they do, but we have some idea that they are really important in communication. So these are like the essential receptors in our cells. So we need to make sure that they're at least not moving as much as we want to. We want it stuck in one area, which is why they're slightly thicker and more ordered. But the big important thing to know about here is like the role of cholesterol. Okay, so cholesterol actually broadens the range of membrane fluidity. So at low temperatures, if we can actually get a cholesterol in there, we can actually prevent some rigidity to an extent. And at high temperatures, our cholesterol can actually increase our melting point to avoid denaturation and maintain still like some fluidity to an extent. Okay. So what cholesterol is doing is basically mitigating the effects of temperature and its effects on the cell membrane, right? So again, with low temperatures, we're going to have more rigidity, right? What cholesterol does is prevents rigidity to an extent, okay? So that we have a broader range of what we can survive with certain temperatures for a cell membrane. At high temperatures, we have more unordered, right? We have a more uh, disordered phase of our cell membrane. So what cholesterol does is bring in more rigidity to make sure we don't denature any of our proteins, and where we can maintain fluidity without it all the phospholipids just floating away because there's nothing attaching to them. So cholesterol is kind of just mitigating. It's kind of the buffer system of cholesterol. Okay. Any questions on cholesterol? I know it's sometimes a little bit harder to grasp what it does. types of transports. There's 6 ones that we're gonna talk about. Our simple diffusion, which is just going down its concentration gradient with no protein. Our facilitated diffusion, which is going down our gradient but with the protein helping. Our primary active transport, which is using ATP to move something against its concentration gradient or electrochemical gradient. And then our secondary is actually using the movement of one ion to feel the movement of another one against its concentration gradient. We have our ion channel, which is just moving ions down its concentration gradient. And then we have our ionophore, which is just a mediated ion transport down its electrochemical gradient. If I remember, our ionophore is the one that we don't talk as much about because it just rarely comes up. But the other 5 are really, really important to know the differences between each. And then we have to talk a little bit about the difference of electrochemical gradient. So chemical gradient is just based on concentration. So chemical gradient is going to go down its concentration base, going from a high concentration to a low one. Our electrical gradient is just going to kind of find that balance between positive and negatives, because positives like our negatives, negatives like our positive. So we just try to match to make sure everybody's happy there. All right, channels and transporters. So these channels and transporters have low activation energy, and they actually are able to remove hydration shells and facilitate solute movement. The channels are going to be passive, okay? So that means they're not using ATP. They're highly specific, and they have infinite ability to transfer ions, okay? So we have a picture of what an ion right here looks like. It's basically a gate, okay? It opens, it closes. That's it. Our transporters actually have a 2-gate system, okay? So you're gonna see that we can actually open one gate first and then open the other gate. And that's how it goes. The big thing is though, transporters do not have that infinite ability to transfer stuff. It has a limit to how much it can transfer. And that's what's showing right here in the graph, right? For our ions, it just keeps going up. This positive slope just keeps going up. For our transporters, we actually have this kind of like, our hyperbolic curve. So it just levels off eventually because we have a max of how much it can do. Okay. So these are the differences between both.
Could you explain why? It doesn't really make sense to me. Like if it's something where it's just one gate, why, and it's got to go with a concentration gradient, why does it not level off at a certain point? And why does the
Well,
pump not just keep going?
For like this kind of this ion channel picture right here doesn't give it justice, because once we open it, it's just a flood basically, because we only have one gate, right? It's only a huge flood, so it just keeps pushing through. It's not like every time one enters, it's going to close until it's open again. No, it's open, it floods in. For a transporter, if we didn't have an extra gate, we could do that too. But because we have 2 gates, it means that we have to open one, close it, and then open another one. Okay. It's like a 2-latch door, like a 2-door system almost to get to one area, which makes it really slow in transporting it. Okay. Because it's 2 different like mechanisms that need to occur for whatever solute to move into the cell. For an ion, it's just open one time and everything floods through.
So is that graph not necessarily for a long time? It's sort of the velocity graph for like one unit coming across. Like the for the top one, it just flows through, but for the bottom one, it comes in and slows down, and then it goes.
Yeah, I think that the graph for the transporter or for the pump here, it's a lot of a it's a better illustration of what's happening. For the ion channel, it's the ion channel is just straight up a trap door. Everything flows through as much as it needs to until it reaches equilibrium. For a transporter, it just takes a long time because we have these 2 alternating gates. And that's why for the ion channel, it can be infinite ability to transfer ions until it reaches the equilibrium. Okay. For our transporter, it's just individually we have a limit to how much we can do because we have 2 gates opening and closing alternatively, and that takes a large amount of time that ion channel doesn't have to deal with. Does that make sense? kind of.
A little bit.
Yeah.
I'll deal with it.
Sorry. Yeah, it's just a little bit weird, but like the thing I always think about is like the ion channel is just straight up open, everything flows. So that's why it's so fast, and it doesn't need to necessarily end because it's just consistently flowing through. The transporter, it's just we have to move these gates all the time, and it just slows everything down. But so lecture 24. So for lecture 24, we first started talking about our glucose transporter. So we actually have a polar core for our glucose, and then 12 transmembrane helices around that one core, okay? We now have a new KM, which is our KT, okay? So the first thing we have to know about a KT is that it has the same properties as KM. So when I ask which sugar does GLUT1 have a higher affinity to, what do you guys say?
Glucose.
Which glucose?
D-glucose.
Exactly, D-glucose, because it has a lower KT, right? Because lower KM means it has a higher affinity. So our D-glucose, right? The one thing the way that I always remember this, especially if we're looking like at L-glucose, remember that our body doesn't really take in L carbohydrates. We take in our D carbohydrates. So that's why we'd have a higher affinity to our D carbohydrates. So now we have GLUT1, 2, 3, and 4. Okay. So you do have to know the role of each one of these glucose transporters in our bodies. But I will say the most important ones are GLUT2 and GLUT4. And that's because they're going to come up in exam 3. So it's better to memorize them right now before you have to memorize it again for exam 3. Our GLUT2 actually has the lowest affinity out of all these glucose transporters. And they actually regulate the removal of excess glucose from our blood. They're mainly going to be found in our livers, kidneys, and to some extent our pancreas. And what they do is that they have such a low affinity that only huge spikes in glucose are going to activate them. Okay. And once they activate, they can do a lot of different things. Okay. They can actually even stimulate insulin production because it's telling our body, yo, we have a lot of glucose circulating and we need to find a way to remove all of them. GLUT4 has a really, really high affinity. It's not the highest affinity, but it has a really, really high affinity. And what's important here is that GLUT4 is actually insulin dependent. So once GLUT2 activates that insulin production, insulin is going to activate GLUT4 to start picking up all those glucose circulating in our blood to our muscles, fats, and hearts. Okay? Any questions on this? So our anti-Porter system, okay? So can anybody tell me why this is an anti-Porter system?
Flowing in flowing in opposite direction.
Exactly. That's an anti-Porter, right? It flows in opposite directions. So you're gonna see right here, this is a really, remember when we talked about for exam 1 about the carbohydrates and the bicarbonate buffer? This is kind of like what's actually happening, right? This is like a level above of what we just talked about. So you're gonna see that as we're breathing, we form carbon dioxide. It enters through our erythrocytes where they can go through our carbonic anhydrase to form bicarbonate and chloride ion. No, sorry, we form bicarbonate and our hydrogen ions. The bicarbonate leaves this respiring tissue while our chloride ion goes inside our erythrocyte or our red blood cell, okay? Our bicarbonate will then go to the lung tissues, go in, our chloride ions goes out, which is again why it's an anti-Porter system. And then our bicarbonate can actually then go through another carbonic anhydrase to form water and carbon dioxide, and then carbon dioxide leaves our erythrocytes through our exhalation, okay? Does that make sense? Any questions on this?
So this reaction, it's going to go in the opposite direction when it's in the lungs. It's sort of like showing us a cycle.
It's just because it's really hard to move gas in our blood, so we need to form it into an ion to move it a lot easier. Which is why we form a bicarbonate ion with our carbon dioxide, and then when we reach that lung, we reconvert that bicarbonate ion back into carbon dioxide, and then it leaves through our lung. So new equations. I know everybody hates it, but these are the new equations that we're going to talk about. The good thing is though, I do have, this is actually an old drawing of mine that I did here, but there are like, this is like the best way that I found to understand how this works. Okay. So this is the question that Xylee gave us, right? It's what is the energy cost of pumping carbon dioxide from the cytosol? Where so cytosol is inside the cell, right? Where its concentration is 1 to the 10⁷ molarity to the extracellular fluid where the concentration is 1 millimolar. Okay, then assume physiological temperature, 37 degrees Celsius, and a membrane potential of -50 microvolts. Okay. So this is a more complicated version, right? Because we have this one, which this is basically telling us our delta G non-standard. And this is just our delta G of our transport, okay? So this is just kind of a derivation of this one. So what you're gonna see right here is that we have our R constant, which is 8.315. We have our 310 Kelvin, which is given to us, ln of our concentration, right? So our C2 is gonna be the concentration that we're going towards, right? Or of the destination we're going to, which is in this case our extracellular fluid, which is 1 millimolar, which is 1 to the 10 to the -3. Then we have our cytosol concentration, which is 1 times 10 to the -7, plus our Z is just representing what charge our ion has. In this case, it's calcium ion, which has a 2+ charge, right? So 2. And then we have our Faraday constant, which is then given to us. And then we have our volts, which is -50 millivolts, okay? And then we just add, right? So we just keep, but the important thing here actually, which is what trips a lot of people, is what to do with the membrane potential, okay? So if we're going from the outside of the cell to the inside, make the membrane potential negative. And if we're going from the inside of the cell to the outside, make that membrane potential positive. Ignore what's in the question because this is just standard membrane potential, but it's not telling us of which direction the reaction is going towards, if that makes sense. So again, if we're going from the outside to the inside, make the membrane potential negative, but then inside to the outside, make membrane potential positive. And that's the best way because everything else is just mainly plug and chug. It's this one area that's kind of confusing, okay? And then you're going to see that in this question, right, we got up 33 delta G. And that means that reaction is unfavorable, right? And that makes sense, right? Always check that delta G to make sure it makes sense. Since we're going from a concentration that has a lower calcium concentration than the outside, that reaction is going to be unfavorable because it's going against its concentration gradient, okay? So last case scenario, right? Just always make sure that your delta G makes sense with a scenario given to you. Does that make sense for everybody? Okay. I'm gonna try to rush through because I know we're 3 minutes out from leaving or from because I know everybody's probably busy. It's a Wednesday, but okay. So we talked about our P-type ATPase for our phospholipids, right? So now we're going to look into a little bit more deeper to it. So these P-type ATPases are for primary active transport, right? So they use ATP. It has 3 different domains that are really, really important. We have our actuator domain, which activates the displacement of a phosphoryl in ADP to allow a new ATP to join. So I like to think of this guy as the recycler, removes everything that we don't need to open up space for things we need. We have our phosphorylation domain, which takes that inorganic phosphate to allow the transport to take place. And then we have our nucleotide binding domain, which is where ATP attaches and then eventually gets dephosphorylated and then causes the conformational change to allow the transport to occur. Okay. So this is another way of our primary active transport. So we have something called our multi-drug transporter, and these actually pumps drugs and toxins out of our cells. These are really, really important when we're getting sick, when we have toxins in our blood, but it's not good when, for example, we're doing cancer treatment, because then our tumors are going to start overexpressing these multi-drug transporters and pumping all those chemotherapy drugs out of the cell to not kill it. Okay. So these are really, really important. The big thing to know about these multi-drug transporters is that we're actually using 2 ATP to pump them out. So that's, I think, the big deal about it. For secondary active transports, as I said, we're just using the movement of an ion down its concentration gradient to pump something out away or against its concentration gradient, right? So it uses the energy of a molecule going down its concentration gradient. The main example I want to say is our sodium glucose symporter. This is really, really important for the absorption of a lot of our macromolecules in our intestines. So as we see here, 2 sodiums and 1 glucose can actually go through to their epithelial cells. And then we actually have another example right here. Actually, sorry, I was looking at the sodium, potassium, ATPase, and that doesn't make sense for this case. But then you're going to see that our glucose goes to our GLUT2 transporter into our blood, okay? So aquaporins, these are transmembrane channels that are great for water flow, okay? So these have actually selective filters. So we have, as you can see, we have arginine, asparagine. Asparagine and histidine are actually going to be the ones preventing proton hopping, and then arginine is going to be the ones preventing ions from ever reaching that aquaporin, because the aquaporins, even though they're small for water to move through, they're also small where ions can sometimes try to get through. But arginine kind of just makes sure that nothing gets through there. Okay, it's kind of like that gate. And then, okay, last ones. I think I made it. Ooh, almost missed it by 1 minute. Channels. So we have 3 different channels. We have our ion channels, voltage gated channels, and our ligand gated channels. For our ion channels, these are the ones that are going to be moving our ions through our cell membrane, right? So they actually have to dehydrate our ions before entering, because since the ions have a charge, they're actually going to have a layer of water around them because the water is attracted to the charge. But we do have helix dipole strips. We have helix dipoles in these ion channels, which actually is going to strip the water from the ion and stabilize the ion as a whole. For our voltage gated ones, these are going to be the ones that respond to changes in the membrane potential of a cell membrane. So you're going to see that a lot in our nerve cells or our neurons, right? So we actually have arginine, which is going to be acting as a voltage sensor for these gates. And then finally, we have our ligand gated channels, which are just open based on a ligand binding to a certain receptor of that channel and then allowing everything to enter. Okay. That was the last slide I'm going to go to. I'm going to stick around for a couple minutes if anybody has any questions, but if not, you guys are free to go. Okay. Again, good luck. Start studying. Your exam's like in a week. So get studying. For the most part, lecture 25 and lecture 26 is just a little bit more detail of what we just learned. But again, the slides are already there. So if you guys just want to look at it to review or something, it's fine with me. But next week, come in with your questions. There are going to be a lot more exam questions coming next Wednesday. So come prepared. Next week, we're going to just drill in some things that I want to make sure you guys are ready for this exam. Okay. So, but if anything, message me. And again, you guys are free to go. Have a good night. Okay.
Hey, are you going to do another practice exam?
Yeah, it's coming. Yeah, it's always comes on the Monday of the week of the exam.
Okay, great.
And then
Thank
I'll
you.
also probably post from last semester's this weekend as well.
That's great. Thank you very much.
No worries.
Thank you.